Redeem Academy.
There's a little bit of a fight that broke out between these two guys. Why? The things people do just to be famous. I can't get it out of my head. Yo, what is up, Science Squirrels? Johnny here. Welcome to Redeem Academy. I'm here to help you get the most out of your education while giving God the glory he deserves. We're at the end of a unit called What is Biology? And the last lesson today is on cell biology. I want you to know that 4 out of 5 dentists agree that you should subscribe to Redeem Academy. And they say if you want the freshest breath, you should pick up a notebook at my website, redeemacademy.org.
I think your breath stinks.
Didn't see that coming, did you? Big question today, what does learning about tiny, complex machines inside of cells tell us about God? All right, we're wrapping up this unit on what is biology by learning about 2 scientists. Their names are Theodore Schwann and Michael Behe. All right, we're starting off with Schwann. He's one of the main contributors to what is now called cell theory. So we're going to look into his history. Check out this cartoon.
Fast forward to the 1800s, where 2 German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodore Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns.
Those were some really good sideburns.
After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the 2 started working on the beginnings of the cell theory.
All right, so let's add some color to Schwann. All right, so there is Schwann, and he was one of the founding fathers of cell biology. Now, cell theory is huge. So middle of the 1800s, these 2 different guys, Schleiden and Schwann, are looking at 2 different kinds of things all the time. Schleiden's always looking at plants. Schwann's always looking at animals. And whenever they look at these things under the microscopes, they discover everything's always made of cells. And they come up with this broad theory that whenever you look at any living thing under a microscope, it's always going to be composed of a fundamental unit, a cell. Turns out, 150 years later, this is proved to be correct. Everything's always made out of cells. And as Christians, we go, yeah, you know why? Because God made everything, and he uses the same design everywhere he goes when he makes living things. Now, science is performed by humans, and humans have egos, and they have desires, and they have disagreements. And so we're going to look a little bit back at this video and see there's a little bit of a fight that broke out between these 2 guys.
Ready for tea?
A bone of contention arose between them as for the last part of the cell theory, that cells come from pre-existing cells. Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence.
Does that sound familiar? Spontaneous generation? So Schleiden, or Schleiden, depending on how you want to say it, he was basically saying spontaneous generation for cells. And Schwann was like, no way, Jose. And so this is a big topic in the 1800s. It would be later with Pasteur and others that they would prove spontaneous generation is not possible. Today, cell theory says cells only come from other cells. Now, later on, after Schwann died, they actually named a cell after him called the Schwann cell. So since we're doing cell biology, I thought, oh, I'll do a little more on Schwann cells since we're talking about cells today. Okay, before I tell you about Schwann cells, I actually need to explain neurons or brain cells, and so we're going to watch this animation. Okay, so we're going to zoom in on this brain, okay, and we see a bunch of brain cells firing, okay, and so we zoom in further and we see that there are electrical signals going across these nerve cells. Now I'm going to back up. I don't usually do this. I usually do the opposite, but I'm going to slow this video down. Okay, so now we're watching this video really slowly. Okay, so this is the end of one neuron, and this is the body of another neuron. Now, as this fast forwards, it zooms in on the end of a nerve. I don't even know if the word end is the right word. We could call it the arm. So this arm of this nerve right here is called an axon. And electrical signals travel down axons, and it's actually those little white dots represent salt. And so those are ions, which is another word for salts, or you've maybe heard them called electrolytes. So it's the movement of ions across the axon membrane that causes electrical impulses to fire through nerves. So this picture right here is some drawings that Schwann made from his microscope that ended up in a book. So you can see figure 8, figure 9, figure 6, figure 7. These are all different tissues that he drew under his microscope. So figure 9 here is his drawing of neurons. And you can see these long sections, like this right here, these are axons. Now scientists today have gotten better at taking pictures of these things. They use fluorescent stains. This is super nerdy and cool, where they actually stain the tissues with things that only shine under a black light. It's literally like from a crime scene movie or something where everything's dark and they're shining black lights, and they'll take photographs of things as they fluoresce under black lights. And so in this picture, the axons are red, the Schwann cells are green, and the nucleus is purple. And so it's not really that easy to tell what you're looking at unless you have a nice cartoon version like this. And so this is your traditional drawing of a neuron, a nerve cell, a brain cell. And Schwann cells are these cells that insulate neurons. I love teaching in my house because I just go get stuff when I'm like, oh yeah, I was gonna explain it with this. So here's some tools, and I'm going to explain insulation now. Okay, so Schwann cells insulate just like plastic insulates a wire. So like here's an old charger from a Samsung phone. We don't have Samsung phones anymore, so I'm going to destroy it. So in the same way that an axon is trying to take a signal from this end and send it to that end, in the same way we're trying to get electricity from this end to charge the phone, the thing going from here to here, we're just trying to get it to travel down the wire. And what we're trying to prevent is the electricity from like grounding out somewhere else. And so let's cut this in half here. Okay, and what's actually in here is mostly plastic, but in the very center, let's try to zoom in here. Okay, you can see in the very center, there's a tiny bit of metal in there. So I'm going to strip it out a little more. Okay, and now you can see we've got a ton of plastic in the wire, and then we've got a second layer of thinner plastic around the wire. And then we actually have a little bit of copper wire right there. So what we call this plastic stuff is insulation. Electricity can only go through the metal. Electricity doesn't go through plastic. And so we call it an insulator because it won't let the electricity go out. It just keeps the electricity in the wire going to where it should go. In the same way with Schwann cells, there's like an electrical current going through the axon, going through the nerve, and these Schwann cells wrap around the nerve and prevent the electrical signal going through the nerve from getting out and interfering. Now your brain has billions of nerves in it, and they're all wrapping around and touching each other. And so it'd be like a trillion electrical cords in a big mess, like back when we played video games when I was a kid. We had a Nintendo 64 and there were 4 controllers, and those cords would get so intertangled with each other, just turn into this giant mess, take you 20 minutes to get them apart when you were going to take your Nintendo 64 to your friend's house. In the same way, all your brain cells are able to be intertwined with each other, and because they're insulated, they don't accidentally send signals to each other that they aren't supposed to send. So this is God's design that humans figured out later for electrical wires. Yeah, we do that. God does it in your brain with Schwann cells. Amazing. Now here's another interesting picture. Schwann cells grow around the wire that we call the axon. So you can see in step 1, the Schwann cell's getting closer, it's wrapping around, and it forms a whole bunch of wraps around the axon. Now, when a person develops a disease called multiple sclerosis, they start to lose the ability to do things, they can't move their body correctly. It's because their Schwann cells are deteriorating. So most of medicine is just figuring out what thing that's supposed to work right isn't working right anymore. Okay, the cell that insulates neurons is called a Schwann cell. And in particular, Schwann focused on animal cells. Hey, shout out to Schleiden. He didn't make my notebook just because I didn't have space. All right, now we're going to be talking about a scientist who is still very much alive today, Michael Behe. Let's add some color to him. All right, Behe is part of the intelligent design movement, like Stephen Meyer, who I mentioned last time. Okay, in this clip, he describes how he sort of became part of the intelligent design movement.
The bacterial flagellum,
The single-cell bacterium.
is literally an outboard motor that bacteria use to swim. It uses a little piece that spins round and around and around and pushes against the water just like a propeller does in an ordinary outboard motor on a boat in our everyday world. And the propeller is attached to a drive shaft, which is attached to a motor, which has clamps holding it in place, and dozens of pieces that are required for it to do its job. It's just a fantastic example of what science has discovered, and that is molecular machines.
The first time Behe saw a diagram of the flagellum, he was captivated.
I look at it and says, wow, that's really fascinating. I wonder how that evolved.
All right, so I've got a big diagram of the flagellar motor. It's from a single-celled bacterium. So we're going to label the parts, and I'll discuss briefly what they do.
Listen up, please!
All right, so here's a bacterium. All right, and here we have a diagram of a flagellum. We're going to look a little deeper, but the first thing you should notice is that this whole structure is embedded in a membrane that has 2 layers, okay? So a membrane, let's go over here. A membrane is a layer around a cell. Now, we're cutting off the edges here and only looking at a portion, but imagine that these continue around the whole cell, okay? So this is the inside of the cell right here, and this is the outside of the cell right here. So this is the inner membrane, outer membrane. Now, there are a bunch of parts to this thing. There's actually like 30 proteins, and I've managed to just slim it down to naming 5 or 6 parts. This thing is called a stator. It's stationary, it doesn't move. So over here we see the stationary part of the motor that allows acid through. It's called a stator. So how this thing actually works is amazing. Imagine a water balloon. I don't think I can even find a picture to represent this idea, but imagine a water balloon that's full of water, and it's really, really full, and then you're pinching the end of it. The water wants to come out. There's a pressure of water that wants to push out the tip of the water balloon, okay? That is going on in between the 2 membranes. So there's 2 membranes. They're like a water balloon that's holding all this pressure. Only it's not water, it's acid. Crazy. So in between the 2 membranes of the bacteria, there's a big buildup of pressure of acid. It's not so much like a physical pressure, it's a chemical pressure. And so it's a pressure of like high positively charged ions, acid ions, that want to push out. So think of the stator as like the nozzle that can let the acid out, and the acid is right here. It's between the 2 membranes, and this picture doesn't do a very good job of showing that. Now, when the acid comes out, it's able to spin this thing, okay? Now, these guys right here are called bushings. Okay, bushings exist in other machines as well, and they act as protective rings or bearings to reduce friction. So bushings are in here to prevent the rod from banging around while it spins. Then we have a rod. This thing's just called the rod. Okay, it's like going to be the spinning part. We have a hook. That's a pretty easy name to remember. And then we have what's called the filament. Okay, so in a car, we call that rod an axle. It transfers the spinning motion to the hook and the filament. That's the rod. And this is what powers the flagellum to spin. It's a flow of acid. Now, the last thing that we need to label is what's called the secretory system. Now, that's a weird name. Secretory system. Okay. And those are these parts right here. They have the word secrete in them because they secrete out all these other parts as this thing actually builds itself. Okay, so that's a lot. Let's watch some videos of how this thing actually functions.
Perhaps the most amazing propulsion system on our entire planet is one that exists in bacteria. It's called the flagellum, a miniature propeller driven by a motor with many distinct mechanical parts, each made of proteins. The flagellum's motor resembles a human-designed rotary engine. It has a universal joint, bushings, a stator, and a rotor. It has a drive shaft and even its own clutch and braking system. In some bacteria, the flagellar motor has been clocked at 100,000 revolutions per minute.
100,000 RPMs. That's 1,666 revolutions per second. Can you imagine driving around in a car and every second the tires spin 1,600 times? Pew! You would be cooking.
The motor is bidirectional and can shift from forward to reverse almost instantaneously. Some scientists suggest it operates at near 100% energy efficiency.
100% energy efficiency. You probably don't know this, but your gasoline engine in your car is about 25% efficient, okay? When you turn on your car and you run the engine to go places, of all the energy that's in the gas, about 75% of it is wasted on heat, just heating. Your engine is really hot. Most of the energy when you burn gas comes out as heat, not as moving the car forward.
All of this is done on a microscopic scale that is hard to imagine. The diameter of the flagellum motor is no more than 5 millionths of a centimeter.
That's a centimeter. Divide that 1,000,000 times and then add 5 of them together. The thing's probably like
25 or 50 or 100 atoms wide.
It's just the tiniest thing you could imagine. It's right there down to the size of those little cars that James Tour was making. All right, enough amazingness of the flagella. So, what does the flagella have to, I mean, it's a little feature inside of a cell. What does it have to do with anything that I want to talk about today? Let me show you.
I look at it and says, wow, that's really fascinating. I wonder how that evolved. Somebody must know this because everybody says Darwin's theory is true. Then I went back and said, well, who does know how this could have evolved? And nobody had anything to say about it. Even hand-waving speculations were hard to find.
Behe began to dig deeper to find an answer.
Darwin's theory says you have to start with something that's working a little bit, and then it will change, mutate a bit, and that helps a little bit, and that'll be selected, and then improve a little bit more and a little bit more until you get the full-fledged system. And I said, well, it does not look like some of these systems can be done that way because they need all of these parts. You know, if you don't have this one, it's not going to work. You can't take any of the parts away. I was kind of sitting at my desk scratching my head. You know, you can't take the parts away. You can't reduce it at all. It's irreducible, and so it's irreducibly complex. I thought, bingo, that encapsulates the problem for Darwin's theory right there.
Okay, did you catch that? So Behe coined a term because he couldn't explain how would this bacterial flagellum evolve. It needs all the parts at once. And he called it irreducibly complex. Let me give you an easier to understand concept of irreducible complexity. This is a much younger Michael Behe. I actually saw this documentary my first year in college, 2003. This is a very old documentary.
Irreducible complexity was coined by Mike Behe in describing these molecular machines. Basically what it says is that you have multi-component parts to any given organelle or system in a cell, all of which are necessary for function. That is, if you remove one part, you lose function of that system.
The idea of irreducible complexity can be illustrated by a familiar, non-biological machine, a mousetrap. The trap is composed of 5 basic pieces: a catch to hold the bait, a strong spring, a thin bent rod called the hammer, a holding bar to secure the hammer in place, and a platform upon which the entire system is mounted. If any one of these parts is missing or defective, the mechanism will not work. All components of this irreducibly complex system must be present simultaneously for the machine to perform its function, catching mice.
Now, in honor of Michael Behe and mousetraps, let's watch somebody slap their tongue with a mousetrap in slow motion. All right, this video is slow-mo, guys. They have a great channel. I love things in slow motion. I don't know if I'm gonna like this one. Oh, why? The things people do just to be famous. But hey, see, this bar right here is called the hammer. That little guy right there is called the catch. That is, I don't know, like this bar, this is called the bar. That's called the hammer. That's called the catch. This is the platform, and you got a spring, and then you got some nails that hold everything down. There you go. Those are the components of a mousetrap. And one more time. Oh, mama pajama! I can't get it out of my head. So anyway, back to Michael Behe. Basically, his argument is you can't evolve a bacterial flagellum because you need to start with something simpler, and you need to evolve and benefit every step of the way. So maybe you don't even have to evolve like for a tail. Like you don't have to evolve for like something that pushes you, but you need to evolve some beneficial thing every step of the way. And there's no explanation for this. Now anyway, back to Michael Behe. We're gonna call him Dr. Behe. He's a biochemistry professor, researcher who was one of the pioneers of the ID movement, and he coined the term irreducible complexity. Irreducible complexity is the idea that certain cellular machines cannot evolve step by step because all of their parts must be present and working together at the same time in order to function. The object that's often used as an example of irreducible complexity is a mousetrap, and the micromotor used as an example of irreducible complexity, although there are very many other examples, is a bacterial flagellum. All right, I don't know if I did the best job explaining that one. There's a whole book on it called Darwin's Black Box. Really great book. I read it in college. There's some great videos out there on YouTube. You can watch about this stuff from the Discovery Institute. Remember, their YouTube channel is Discovery Science. All right, big question. What does learning about tiny, complex machines inside of cells tell us about God? All right, hot take here. I've never heard anybody talk about it like this, but this is my opinion. The universe is a desert of nothing, as far as we can tell. It's like God took a shotgun of galaxies and just went, and it's all a bunch of dirt out there in outer space of planets and suns. And we can't find any evidence that there's anything else alive out there. There's this thing called SETI, the Search for Extraterrestrial Intelligence. I think SETI ends in an I. Fact check me on that. But, so when we look out, we don't see anything really saying intelligence out there. But when we look down and into cells, things just get more and more complicated and complex. Last lesson was specified information. Irreducible complexity can also be listed as specified complexity. And so we see so much complexity in cells. We look down, we see God spent time thinking about complexity and designing complexity into the teeny tiniest little packages. Earth is a special place. God spent time making stuff here. And I think it speaks that God doesn't care about size. He made a giant universe, I think, to speak and say, I'm big and powerful. But then he's taken so much time to make really intricate things to say like, yeah, I can make big stuff, but I can make small stuff too. And I think it says something about how size is not the thing that defines who God is. God is fundamentally outside of the universe, and he's outside of size. He is more concerned with information, or more concerned with intelligence, more concerned with other things besides just size. When Bill Nye has this take where he says, I'm a speck on a speck, you know, and he tries to describe how insignificant humans are on the earth and try to say, you know, something to the effect that, God doesn't exist and he certainly doesn't care about us. No, I don't think so, bro. God is not concerned about the size of us in comparison to the size of the universe. The complexity of design inside of cells and the complexity of us on this planet, we're different, we're special. God has something special in store for us on this earth because of the uniqueness of who we are on this planet. Hey, I hope you've enjoyed this introductory unit of what is biology. Tell me what has been most interesting for you in this unit. Tell me what you'd like me to do differently. I've got 9 more units to go in my biology curriculum, so I'd love to hear what's working, what should I do differently. Give me some feedback. I've never taught biology online before. Give me some feedback. I hope you're here having a good time, and if you are still here, thank you so much. Have a great day. Coming up next is taxonomy. I just finished the unit, and so I'll be doing 8 lessons on taxonomy. Stick with me and see what that's all about. Have a great day.